Good morning. What a wonderful turnout for this great lecture to come. I'm Matthew Wilson, the Anatomy and Pathology Ocular Oncology Trustee, and it's with great pleasure to provide the history of the Mildred Weisenfeld Award for Excellence in Ophthalmology. The award was established as a tribute to Mildred Weisenfeld's outstanding contributions to the field, which included the founding for Fight for Sight. In 1946, she believed that by encouraging research and researchers, that result would have a greater impact. The award is presented to an individual in recognition of distinguished scholarly contributions to the clinical practice of ophthalmology. The 2024 Mildred Weisenfeld Award is presented to Martine J. Jager, MD, PhD, Professor of Ophthalmology at the Leiden University Medical Center in the Netherlands. Her work has focused on the role of the immune system in corneal transplantation and ocular oncology. It is with great pleasure to present this year's award to Dr. Jager. Please come forward.
Thank you so much. Thank you.
I am now pleased to introduce Dr. Klaus Kurseven, Director, Department of Ophthalmology, University of Cologne, who will review Dr. Jager's achievements and introduce her lecture.
Yeah, dear colleagues, ladies and gentlemen, good morning. Dear Martine, it's my great pleasure to introduce the 2024 Mildred Weisenfeld Award recipient, Professor Martine Jager from Leiden in the Netherlands. You already heard that the Weisenfeld Award is about honoring excellence in ophthalmology, so that's about Martine Jager. In addition, you also heard that the award is also a moment for us to pay tribute to Mildred Weisenfeld for her outstanding contributions to our field, basically single-handedly establishing what is now known Fight for Sight and also being a key player in developing the National Eye Institute. And it's even more impressive if we consider that she was losing her sight due to RP during this time and had no specific scientific training. And she did all this as a, so to say, one-woman campaign. That's a big difference to our awardee here today. Martine Jager was privileged to always have her twin sister on board, supporting her from very early on to up to now, Johanna Jager. And if you look at these early pictures, you can see already a very early interest in biology and science, investigations, which later on diverted to sports and also a little bit to art. So let me formally say a few words about Martine Jager. Her academic journey started in 1974 at Leiden University, where she earned her MD and later on her PhD. As an ophthalmologist, she excelled in translational research in ocular immunology, with a special focus on oncology and corneal diseases. Her academic positions included roles in Leiden, the Netherlands Royal Academy of Arts and Sciences, and also the Basque Palmer Eye Institute. Coming back from the US, she established her own lab in 1992, where she managed, besides being a clinician, to contribute significantly to ocular immunology. Just to give you a few spots of her significant and excellent contributions to ocular immunology, that is recognizing, for example, the importance of inflammation in uveal melanoma, describing new key cell lines, describing the role of GP100 in uveal melanoma cells, and recognizing the importance of an inflammatory phenotype and also eye color as a bad prognostic factor. In addition, she also contributed to the field of cornea, being key in developing the corneal registry in the Netherlands, and also involved in being in the development of an artificial cornea. But besides all this, and on top of that, I think very important aspect of Martine Jager is her being a role model and a mentor for numerous students and PhD students. You can see here pictures from a very recent PhD defense, or after a recent PhD defense, where I had the pleasure to be participating. She always had a very strong focus on supporting international students, minority students, and also female scientists. Here I see some of the pictures. Some of the PhD students and postdocs are already here in the room. Another, I think, very important aspect is her role as a leader, and I would say a networker for the good. So not only networking for her own purpose, but for the greater benefit of ophthalmology, for academic ophthalmology. And she used basically her numerous leadership positions to do that. And just to give you an example here, as the Secretary General of the Academia Ophthalmologica Internationalis, in several European Union projects, coast actions, for example, this picture here is on biocornea development. And obviously, foremost, her role as the leader and networker for ARVO, being the first non-American president of ARVO in 27 to 28. For those of you who know Martine, she is a fighter for things she has recognized as being worth pursuing, a fighter against all odds, and for that she was honored with the knighthood of the Lion of the Netherlands. You can see here the King and the Queen of the Netherlands. So with this, I would like to thank you, Martine, also personally for supporting us, for accepting a new role as a visiting professor at our department in Cologne, in Germany. I wish you to stay curious, as you have always been. And now I would like to give a big applause to our 2024 Mildred Weisenfeld, our dear, and ask you to come up to the podium, please.
Thank you very much, Klaus. It seems only a few years ago that we were seeing each other when you were the winner of the Cora Verhagen Award for Excellence for a Young Person in Immunology, and I had the pleasure of giving you the plaque. And now you are head of the department in Cologne, and if that is not important enough, he managed to get his team together in a fabulous way and raise 50 million euros to study inflammation of the eye. So that's a great way to stimulate care of eye diseases. Well, my career looks fabulous. However, for all those who ever get into trouble, it's not always easy. When I had been visiting at Harvard and saw that Joe Miller became chair of the department there, I started wondering, could I at some time become professor? And I was old enough to do that. So I went to our administrator and said, maybe it's a good idea if I take a leadership course. I want to get up in the ranks. And I looked above me. There were no women. There was not even one female clinical professor in our whole hospital. And the man looked at me and said, "Ma, you are not worthy of such a thing. You will never make professor." Well, it changed, but it was very, very upsetting. In that same year, I was candidate to run for trustee of ARVO. And I was elected. And then a couple of years later, ARVO elected me to be its president. And then Leiden University said, maybe we were not so smart. And ARVO had put me on a leadership course at the American Academy, and that was absolutely fabulous and prepared me for a later career. So ARVO has meant very, very much for me in my life. And that you are all here is fabulous. You come to Seattle to this great meeting to learn and to meet each other. But organizing a meeting is very hard work. And when you organize something, you only get comments. And I want you all to stand up. Good exercise this morning. Please stand up and give ARVO leadership, Iris Rush and Lancy Cohen, a big round of applause for organizing all this. So, Congratulations, congratulations, Iris and Nancy, on a great meeting again. It's fabulous here with so many people. So it is not a surprise that the Weisenfeld lecture is here at ARVO, and I thank the awards committee for asking me to give this lecture. All great organizations like Fight for Sight and Champalimaud are here to celebrate with you. And I have one financial thing to report, a Health Holland grant. Mildred Weisenfeld came up with one very important thing that has not been mentioned about her yet. Instead of using charitable contributions to help people get help with their blindness, she decided it was better to cure the disease and to raise funds for that. Many of you have benefited from money from Fight for Sight, and she urged Congress to add blindness to the Institute of Neurology, and the National Eye Institute was created. Just one woman who decided that money had to be used to cure diseases. Well, Weisenfeld Awards, there have been many, and up to 2015, there were no women in it. Fabulous men, fabulous men. And I have to point out one, Paul de Jong, who was leading eye research in the Netherlands in the previous century. Now, when I started out, ocular immunology was not a thing. Innate immunology was not considered important at all. Now, this year, you can see immunology, inflammation everywhere. Whether it is in brain health or gut health, dietary and genetic factors influence inflammatory bowel disease. You cannot read any newspaper or it's about inflammation. And I recently came across this. The market value of Novo Nordisk has surpassed that of Tesla, even Tesla. The stock rose because of the effect of Ozempic and Wegovy. Why is this important? It's a drug for weight loss. These drugs influence weight loss through their effect on inflammation. And not many people know it, but statins, used to fight vessel disease, also affect inflammation. That was not relevant then because inflammation was not relevant. But these drugs influence inflammation. And these blockbuster anti-obesity drugs, I just learned that socialites in New York use these drugs just to be even skinnier. But it raises hope that it can be used to treat many diseases. Alzheimer, Parkinson's. Those diseases also suffer through inflammation, brain inflammation. My question, if these drugs affect inflammation, may they help to prevent or treat eye diseases? And maybe a couple of years from now, when the side effects have decreased, instead of taking a statin every day after 50, people will use one of these to reduce the inflammation in their body. Aging affects inflammation, it increases it. The work I'm describing will be by many of these people. It has been for me an extreme pleasure, and it still is, to support young people who want to have a career in science. And several of them continue in research. Most of them chose another route and are fantastic cataract surgeons, which is, I have had cataract surgery, it's fantastic. Very useful. So why do I do research? What are macrophages? How about cornea transplants? And inflammation plays a role in glaucoma. When I was a resident, the only thing that was important in glaucoma was eye pressure. And then came normal pressure glaucoma. That was, when I was a second-year resident, that was a very big thing that existed. Before it was just pressure, you press on the nerves and they die. That was it. Then uveal melanoma, and what can we do to change innate immunology? As a third-year student, I had the great pleasure of studying for a while at the University of Edinburgh, and I was caught by the research virus. Leiden is a lovely city and very focused on translational research. So when I graduated, I wanted to do a PhD, and the really important laboratory in Leiden was the HLA laboratory of Jon van Rood. So I applied, and he took me in for that. And I studied a subject nobody else was interested in, monocytes. I stumbled upon them, and I found them lovely little creatures that I could look at. And so I did my PhD on that. But somehow, ophthalmology attracted me, in spite of my father being an eye doctor. I still wanted to go into that field. But before starting a practice, I wanted to learn more. And I went over to the United States. There was a world-famous immunologist, always here at ARVO, talking to friends, being in the hallway. Everybody ran up to him. He knew everybody. Wayne Stryline. And together with Jerry Niederkorn, they really established the immunology of the anterior segment of the eye. And he introduced a lot about eye tolerance. So I joined there, and there was a simple project, how to look at mice that had herpetic infections. One would scratch 50% of the mice on their eye. That would bring in antigen-presenting cells. And the question was, does that affect the herpetic infection? If the cornea had more antigen-presenting cells after the scratching and then had the virus go into the cornea, then that mouse would develop much more severe corneal inflammation. The mouse might even die of brain herpetic response. But it was the immune system that caused it. So the presence of antigen-presenting cells in the cornea make the patient worse. As a clinician, I have treated very many herpetic patients. And the Weisenfeld Award is for improving patient care. Just this thing that I saw the mice and realized how there was an interaction between the viral infections and the immune system has helped me treat patients over the next many, many years. It's exactly the same. And what Wayne also did, he introduced me to ARVO. And it is very much fun that the people that I met in Miami in 1989 are still friends. If I look around here and I see Sally Atherton and Andy Taylor and Bruce Xander, we stayed friends forever. So for you who are young researchers, people that you meet when you are a resident or a postdoctoral fellow, you may meet the same people again 30 years from now. So make sure you make friends and not enemies. And I love that we remained friends for such a very long time, very important in my life. And as I said, here with Joanne Engel and Sally Atherton when I became president. And please notice, among the members of the board, Sally and Joanne are not members of the, not the elected board members. They're all men. That has changed over time, but that was a very active procedure in ARVO, for which I compliment ARVO for doing that over all these years. And I think especially Justine Smith is very responsible for doing this. Great job, Justine. So after working in the US, I went back to the Netherlands and joined the staff at the LUMC as a cornea doctor. And I realized I had to learn more. And having been in Miami, I was accepted as a cornea fellow at the Bascom Palmer Eye Institute. Beautiful, fantastic place. And I learned very much about how to treat corneas, how to do a cornea transplant. I also realized that you may do a beautiful transplant, but it may not go right. You can have a rejection, just one little knot that loosens up, and you get a rejection. What does that one loose knot do? Residents are taught, if you see a loose suture, remove it immediately. It brings in antigen-presenting cells. So those antigen-presenting cells, beautiful picture from the Reza Dana group. You see the cornea. In the cornea of a cornea that is irritated, you have antigen-presenting cells. They go to the lymphatic vessels, they go to the draining lymph nodes. There they trigger T cells, they go into a blood vessel, and then they go to the cornea, and they reject it. And this is the whole process. For those who are not immunologists, this is the basis of almost everything immunological. You first start out with an antigen-presenting cell, the innate immunology, and then you go to the T cells. But for the first 30 years of my career, the antigen-presenting cells and the innate immune system were not really considered important. People had identified T cells, and that was the real thing. And there's a reason now for the importance of these. The current treatments with monoclonal antibodies for many autoimmune diseases and for cancer, they all target the T cells. So very important cell. But I happen to be interested in the other set. And I already showed a picture of Jerry Niederkorn. As I had benefited very greatly from traveling abroad, I helped students who wanted to go abroad. And it's very easy. You write an email or a letter to a friend elsewhere that you have met at ARVO, and you ask the question, would you have a position for this student? So one of our most enthusiastic students immediately said, I want to go to America. He did. And he worked with Jerry Niederkorn in Dallas. And Jerry also was very interested in the innate immune system. What they did was they got rid of all the macrophages. Then you don't get this whole system of antigen presentation and T cells. And when you do that in herpetic disease, you don't get inflammation anymore. But here, there was another effect. The role of macrophages. When you got rid of the macrophages, the eye became completely destroyed if you put acanthamoeba on that eye. This is the other role of macrophages. Macrophage means big eater. Tap, 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 tap. They're like Pac-Mans. And what they do is they eat danger. They can eat bad agents that are present in any tissue. That's the first line of defense. You get a piece of wood in your hand, then the macrophages and the neutrophils run in to try to eat that and get rid of it. That's what they did with acanthamoeba. They would remove the acanthamoeba one by one and save the cornea. So completely different role for macrophages. Now, having been in Miami, something happened. Wayne Stryline moved to Boston from Miami. And I had been in the Netherlands for 7, 8 years, and I was wondering, what is it like? And again, a quick message, and Wayne said, come over, become visiting professor and have fun here and learn what we are doing. So Andy Taylor had moved to Boston, and Bruce Xander had moved to Boston. And quite soon, I met a neuroscientist, Dongfeng Chen. And she was really studying how nerves grow. And also, she had a model for glaucoma. Now, you think, what has glaucoma to do with the immune system? It was something I was wondering about too. But she said, no, we have found something very interesting. Heat shock proteins play a role in glaucoma. And we started chatting, just chatting and exchanging ideas, and then wondered, how about the role of the antigen-presenting cells? Are they important? What do they do? And one of our students, Ganvu, a brilliant Dutch medical student, decided she wanted to get a PhD and wanted to go. She said immediately, "Oh, Boston, that's a great city. I want to go there." So she did. And as you can see, there are very many people involved in a study like this. And they set up a model they had a model for glaucoma. You put beads, you put microbeads in the anterior chamber of a mouse eye, and it blocks the trabecular meshwork so that fluid no longer goes into the Schlemm's canal. And you get an acute raise in eye pressure. Here is the eye pressure curve. This is without the microbeads, and the top line where you see the pressure increase, only up to about 23, 24 millimeters mercury. You see that the pressure goes nicely up and then it goes down again. This leads to loss of the retinal ganglion cells and the axons. This is the retinal ganglion cell loss. You see the increase. Here is the pressure, and you see the increase. Get 2 weeks, 4 weeks, 8 weeks. You get more and more loss of ganglion cells. All as you all expect when you have been working in glaucoma research. But in addition to that, she went deeper and studied the antigen-presenting cells, which are present here. And this is the influx of antigen-presenting cells and the influx of T cells. You first get the influx of the antigen-presenting cells. They pick up things that usually are not there, the pressure, killed cells, damaged cells, and released heat shock proteins, which are not supposed to be there. And it was so much that you don't get suppression, the work of Stryline, but you get immunization. The macrophages go to the lymph nodes. In the lymph nodes, they present the new antigen heat shock proteins to the whole body. You get T cells. The T cells go back to the eye, and they remove the damaged, but not yet dead, other ganglion cells. So if you take a mouse without a good immune system, the damage stops after 2 weeks. If you take a mouse with a fully functioning immune system, T cells and antibodies, you then kill a lot of the other ganglion cells. So it's a two-layered thing. You have the pressure that damages and kills ganglion cells, and then you get the immune system that comes in, the antigen-presenting cells, and subsequently the T cells. And they have shown that you find exactly these same T cells in patients with glaucoma. Here is the system that they have described. The words macrophage and microglia can often be used can be exchanged for each other quite in the retinal tissue. So the microglia or macrophages acquire the antigen, then they present it to the T cells. You get a lot more T cells. They go back and they then remove the already injured cells. So this means glaucoma is not independent of pressure, but it is a combined thing, pressure and damage. And maybe there are other things as well. So I'm a cornea doctor. Why ever would I look at tumors? There was a fantastic session yesterday on inflammation. At this ARVO, wherever you go, you see posters about inflammation. I don't think it's the Ozempic, but it's the time. People are now interested in what happens. But many years ago, yesterday there was a presentation about what type of cells do you find in the cornea. Now, I wanted to have monoclonal antibodies that at that time were difficult to get. And the professor of pathology had the most fabulous set of monoclonal antibodies. So I went to him and said, I want to study the cornea. Oh, great, that's very nice. You can get my monoclonal antibodies. But in exchange, I want from you tumors, human tumors, to study. Eye melanoma is intriguing, and please help me get some. And then the next question was, well, you are staining corneas, perhaps you can help with the tumors as well. And then, well, you have done the staining, maybe you can check them. Oh, you checked them, maybe you can write the paper. Okay. So that was my first paper in oncology, and it led to a lifelong interest in oncology. I also, at the same time, had the patient, Mrs. A, and she came in for glasses. And when I looked in her eye, I thought, that's weird. I see something, it's not macular degeneration. I had never seen it before. And I found out it was an eye melanoma. And then I had to read up on it. And she was already in her 70s. But I learned that that is a very bad tumor. And we had in Leiden a very good translational research department with very many good facilities for mouse experiments. I had learned in Wayne Strijlijn's lab that mice can tell you things that otherwise you don't know. And we had another very good student, Longley, and we decided to repeat the type of experiments that Van Klink had done on acanthamoeba. Get rid of all the macrophages and then see what happens to a tumor. Nobody cared about the macrophages, but we had this technique, the same technique that had been used by Niederkorn. So we achieved macrophage depletion by injecting liposomes. I said they are Pac-Mans, so all the macrophages start eating the liposomes and then they die. So we got rid of all the macrophages in the eye. And there was a paper by Kelly and Epte who describes that the age of mice influences macrophage behavior. So we got different mice, and we just wondered. I'm not going into AMD. Everybody knows that inflammation is important there. But in parallel, we wanted to know what the macrophages were doing in this model. In order to get a tumor, you must have blood vessels. Otherwise, the tumor will not get enough oxygen. So we thought of a parallel between AMD and tumors. Let's see what happens when we deplete macrophages. We had a batch of nice, strong young mice, and we put the tumor in their eye, and nothing happened at all. Just tumors grew, filled the whole anterior chamber, and that was not the result we expected. But in red, you can see that Kelly and Epte described that the age of mice influences macrophage behavior. So we also had these very, very old mice, 2 years old. And we did the same experiment in the old mice. So in the young mice, no difference. Now, macrophage depletion of the eye in aged mice. Now, here are two curves. Blue, regular old mice, you put a tumor in the eye, it grows. 9 out of 10 mice get a tumor. But now, in the old mice that did not have macrophages, it stopped. The absence of macrophages completely stopped tumor growth. So when mice age, macrophages change. And there is terminology for the different types of macrophages. You have M1 macrophages. That's the one that are Pac-Mans. They are important in young people. They protect young people against all types of infections. And you have M2 macrophages. M2 macrophages are pro-angiogenic. And there are also here beautiful papers by Heping Xu, Belfast, that when you age, your macrophages change. And we found that here too. In aging, your macrophages change. That is probably why things like Ozempic will work against aging. They will affect the macrophages when people age. So we found this difference in 2007 between old mice and young mice, that it had an effect on tumor growth. So for this part, I will go more into human tumors. Macrophages have different effects. They help to fight acanthamoeba, They worsen corneal inflammation in hepatic eye disease. They stimulate cornea graft rejection. They are known to work in AMD and stimulate inflammation. Michel Madigan and Jan Provis in Australia did a lot of work on that. They help to increase pressure-related damage, and they are necessary for tumor growth in old mice. Now, people think immune responses may fight cancer. That also is an old adagium. I forgot to say one thing about the work on the glaucoma. When I pointed out Paul de Jong, we sent him the paper that everybody was rejecting our paper on the eye pressure and the immunology and said, stupid, it's all pressure. And then finally, Dongfeng managed to get it published in Nature Communications. And I sent a copy to Paul de Jong, the main researcher at that time in the Netherlands. And he said, this is a paradigm-changing paper. Best paper on glaucoma in 50 years, because it has to you have to change your idea about the disease. And I think that is true for all of this. All of a sudden, innate immunology becomes important. It was not before, it was T cells. Now, this is important. But the same happened to tumors. Immunology was not important in tumors, and all of a sudden, it became important. These are eye melanomas, different types. And it's a malignant tumor in the eye. It is quite rare, but especially in adults, you only start to get this in Western countries, blue-eyed people, after 60. In Asia, it's a different disease. People get it already in their 40s. The mortality rate, 50% of the patients will die of it. And as yet, there is one treatment, tebentafusp, that delays death, but there is no treatment if a patient gets liver metastases to cure them. So research for this disease is still very challenging, and anybody interested, you can follow a pathway in this and try to help patients be cured. It spreads through the blood. For that, you need a blood vessel in the tumor in the eye in order to get out of the eye and go to the liver. If you want to know more about blood vessels in these tumors, look up Hans Grosniklau's paper for the Jackson lecture a few years ago. Beautiful paper on the blood vessels. So it goes to the liver and you get liver mets. Now, just an illustration how interesting it is to look at macrophages. Patient came in. This patient had been treated for melanoma in the eye. The treatment of the primary tumor goes very well. Usually, proton beam, radioactive plaque goes very well. Leiden is a very big, there are two centers in the Netherlands, Rotterdam and Leiden is very big on treating patients with this disease. We have about 300 patients a year in the Netherlands. So that goes well. But this patient had been treated, came back, and had these black spots on her eye. And it was, oh, help. To get black spots when you have had an eye melanoma in your eye, that is very bad. But a biopsy was taken by my colleagues, and what happened was these were macrophages. Macrophages had, as little Pac-Mans, taken up pieces of the tumor when it had been irradiated, and they came out with the along nerves and just happened to collect at the exit of a blood vessel on the ocular surface. So everybody sighed in relief, and the patient happily went home. But that is often not the case. This is a histology picture. Excuse me. This is a histology of a tumor. Blue are the nuclei and red are macrophages. And this tumor is loaded with macrophages. These macrophages are CD68 positive, CD163 positive, and that's a marker for the M2 pro-angiogenic macrophages. The ones that are increased in aging and are helping to create blood vessels in all types of models. But we studied it in eye melanoma, and there are two curves here. In the Netherlands, we have perfect registries. Anybody is interested in patient data, we register everything from birth to death, like Denmark. So we can follow what happens to our patients for a very, very long time. And here on the top curve, you see the survival curve of patients with a few macrophages, and down here with many macrophages. If you have many macrophages in the tumor, that is not good. And there's one important chromosome and a gene. It's the BAP1 gene. If you lose this gene, then your survival is very bad. It goes down to a 5-year survival of only 40%. And increased risk of death. So everything is compared to the presence of this BAP1 gene, whether it's expressed or not. And Dr. Gaskin looked for the amount of macrophages in BAP positive and BAP negative tumors. These are the numbers of macrophages. If this one gene is no longer expressed, it's gone, then you find very many macrophages in the tumor. You also find very many T cells. So the tumors that are bad, that kill the patient, they have very many T cells and very many macrophages. The opposite of what you expect. You expect that these cells will help protect the patient. They don't. They do the opposite. And it seems that they even help to suppress immune responses. So the absence of the mutation in the BAP1 gene, loss of expression, leads to a lot of inflammation in the tumor. And is that relevant for blood vessels, the way how a tumor cell gets out of the eye? Yes. If you look at the BAP1 negative tumors, you can see that these patients who have these tumors have many more blood vessels in their tumor. And there is an association. The higher the number of macrophages, the more blood vessels there are in the tumor. So we came up with this process. You have the mutation in the BAP1 gene, and you lose also the other chromosome where the BAP1 is located. You get, which is being used in the US for prognostication, a specific RNA pattern. HIF1 alpha goes up, macrophages stream in, you get inflammation in the tumor, which leads to blood vessels. These are all M2 macrophages and immune suppression, and that subsequently leads to metastasis formation. So I've spoken about all these roles from macrophages and their importance. Is there anything we can do? In aging, they increase, and what one really needs is a miracle formula. Not only for the tumor, but probably for the whole body. And oil of old age, probably good industrial strength, and, well, it's not yet available, but we had a collaboration with a company, Aura, in Boston. And one of my colleagues was working on nanoparticles to stimulate the immune response. And what is Aura developing and now doing in different trials? They use a particle that contains on the surface heparan sulfate. It comes from human papillomavirus, and it has a specific attraction for malignant cells. My colleague Sen Ma, who's here in the audience, he showed that blood vessels do not like, these particles do not like blood vessels, and it had been shown they don't like normal cells, but they love to attach to tumor cells. So Aura put a photosensitizer in these little spheres, and then these spheres, these spheres then can bind to the tumor. You get the tumor cells full of the the phthalocyanin-loaded dye, and that will lead to direct killing of the tumor cells, but it also leads to activation of antigen-presenting cells. and it leads to the expression of damage-associated proteins. They stimulate the whole pathway of immune responses. And they make T cells all of a sudden respond instead of not responding. This is being used to treat in trials small melanomas and dangerous nevi. That is currently going on. But we wanted to know, is this all true in our mouse models? And we found in regular mouse models, tumors on the skin, that it works. You can kill tumors and you can get an immune response. But people would often say, oh, but photosensitizers don't work in pigmented tumors. Don't try it, it doesn't work. And we had availability of 2 cell lines that one that was pigmented and one that was not pigmented. So we could test the hypothesis that whether this treatment would work in pigmented or non-pigmented tumors. And we set out to do that. You can see here the pigmented cell line and the non-pigmented cell line. It's available for all types of studies if you are interested. And I think it's already in Cologne. So yellow is killing, or yellow-orange, and the higher the dose, whether it's the pigmented or non-pigmented cell line, or the amount of light that you put on it, these cells get equally killed. Excellent. It worked very well on pigmented and non-pigmented cells. But we were also studying the effect on the immune response. And then we found something that we had not expected at all. I mentioned there are 2 types of macrophages. The good guys, the M1s, the little pigments that remove infectious agents, and the M2 macrophages that stimulate blood vessels. And when Sen looked at the different mice, he could see that when Belsar had been used on the tumor, then the amount of M1 macrophages in the pigmented and the non-pigmented mice, in those tumors, the good macrophages increased. And the bad macrophages, the pro-angiogenic ones, went down. This was completely unexpected. But this type of treatment, where you affect with a photosensitizer and a drug the cells in a tissue, clearly affects macrophages. And that is very interesting and intriguing and worthy of more studies, whether this is also true in other tumors. We are now working on conjunctival melanomas. So this Belsar and light activation induces cancer cell-directed cytotoxicity. It brings in good M1 macrophages and was independent of pigmentation. The macrophages are in the center of very many different diseases. I mentioned working glaucoma. When you age, you get more of the bad M2 macrophages, which may be related to uveal melanomas developing in people over 60. And one would like to have treatments, the oil of old age. With corona, there was an incredible stimulation to study immune responses, and many therapeutic targets, CSF receptor signaling, are related to macrophages, and hopefully from this terrible epidemic, new treatments may develop. Global collaboration in all of this is essential. The best studies that I have published were all through global collaborations. And global collaborations is people. It is you coming to ARVO, talking to each other, meeting other people, and then to come up with new ideas. So why to do research? To do something useful, advance the knowledge, help patients for whom there is no treatment, learn new things. It's exciting when you find something new, and you make friends that may last a lifetime. And it is great fun to help others. I also love to travel, as you could see in the pictures that Klaus showed. And coming here to Seattle is a very great treat. And I have had the pleasure of meeting many friends here and helping many of my students to get to this fantastic meeting. And as Mildred Weisenfeld already knew, by doing research, we can understand eye diseases and treat our patients different than what we did before. Mrs. Weisenfeld found out she was losing sight, and now people are using gene therapy to make progress and help new generations of kids to grow up seeing. And I thank the LUMC team very much for allowing me to do studies and to travel abroad, and for many years not see patients and be able to come to ARVO. And thank you for this fantastic award and for all of you being here this morning and listening to my enthusiastic talk about innate immunology. And thank you for your attention, and I wish the next speaker a great success. Thank you very much for your attention.